This is part 2 of the Chapter 7 lecture notes. Okay, so let me make this bigger. Where I left off was we were talking about the striations within the sarcomere. And just a reminder, the sarcomere is a unit, a contractile unit of the muscle fiber. So there's a lot of sarcomeres that run down the length of a muscle fiber, but they all are composed of the same components. They're all composed of Z lines, N lines, A bands, I bands, and H bands. And the A bands are the dark bands, the I bands are the light bands, because they only contain the thin filaments or actin. The I band also includes the Z line, and I'll show you a little bit on the picture in a minute. But the word light, if you put a capital I in there, I'm not going to write it again like I did dark, but light has an I in it. That's how you can remember the I bands are light, and dark has an A, so that's how you can remember the A bands are dark. And the H band is composed of only thick filaments when the fiber is relaxed, and it does include the M line. Okay, so let's go back to that picture and look at this stuff again. All right, so a sarcomere extends from Z line to Z line. And you can tell that there's, like, after this sarcomere, from here to here, there's another one that's gonna be connected to the right, and then there's another one connected to the left. So on the ends of the sarcomeres is always the light bands or actin filaments. So you'll see actin filaments or light striations on either side of the Z line. Okay, so the M line is in the middle of the sarcomere. The Z lines are where a sarcomere is connected to another sarcomere. So they're found only within actin, only within the thin filaments. And then the A band consists of, it's actually the length of the myosin filaments. There is some overlap with actin, but because the myosin is heavier and darker, it's the thick filaments. That's why you have the dark colored striation in the A bands. And then the I bands are only actin, so they're going to be lighter. The H band is the area where there's only myosin. There's only myosin, and it includes the M line. Okay. For the thin filament, just make sure that you understand that. Let me get my pointer. Actin, or the thin filaments, contain active sites. And these are places where myosin is going to interact or bind with actin. The myosin has to bind with actin in order to pull on it and cause a contraction. So when the muscle is at rest, when the sarcomere is at rest, those active sites are covered up and myosin can't bind with them. They're covered up with tropomyosin, and then the molecule troponin holds the tropomyosin in place. I guess I don't really like this picture that well, but okay, so this is a strand of actin, which you're looking at right here, and the tropomyosin is this rope that kind of covers over the active sites. The active sites are yellow on an orange molecule. The yellow part is the active site. So when the sarcomere is at rest, when it's not contracting, tropomyosin covers those active sites so the myosin head cannot attach to them and bind. And troponin is a molecule, which is brown, that holds the tropomyosin in place. So if the troponin is ever moved, then the tropomyosin moves off of the active sites. And we're going to talk about that because that's one of the steps of a contraction, of a muscle contraction. Okay, the explanation for sarcomere contraction is called the sliding filament theory. And this is what it looks like. Okay, so this is a relaxed sarcomere. Notice the H band and how wide it is. Okay, then a sarcomere that is contracting, what will happen is the actin filaments will move closer together. And so the H band will become smaller. So basically, the myosin will attach to the actin and pull on it and move the actin closer, move the actin fibers closer to each other. I'd love to show you a picture of this. I don't see a good picture. Okay, we'll just have to talk about it. So the mechanism for the sliding filament theory is that the myosin heads of the thick filaments bind to the active sites. And when they bind, that's called a cross bridge. And once they bind to the active sites on actin, they pull the actin toward the center of the sarcomere. And eventually, you don't want a muscle to be contracted forever, so eventually it's going to have, those cross bridges are going to have to detach. That is where ATP comes in. ATP is used to detach the myosin from the actin, and then the cycle repeats. All right, basic structure of a sarcomere, just make sure you understand that it's a contractile unit of muscle, and it extends from Z line to Z line. The M line is the middle of the sarcomere. The I bands are the light bands composed of actin, and the A bands are the dark bands composed of myosin with some overlap of actin. The length of the A band is the length of the myosin filaments. And then we have, the H band is the area of the A band that is only myosin, it's composed of only myosin. The H band actually shortens when the sarcomere contracts. Why do skeletal muscle fibers appear striated? Because they're composed of alternating light and dark bands. And where would you expect the greatest concentration of calcium ions to be in a resting skeletal muscle fiber? And that answer is in the sarcoplasmic reticulum, within the, remember we talked about the triad is a T tubule with, I've lost the word, terminal cisternae on either side. So the greatest concentration in a resting skeletal muscle fiber should be in the terminal cisternae because they store the calcium ions. And we're going to talk about how the role that the calcium ions play in a muscle contraction. Okay, so for a muscle to contract, there has to be some interaction with a nerve. And we call this the neuromuscular junction, where the axon of a neuron or a nerve fiber interacts and comes very close to the muscle fiber. The neuron in this case is called a motor neuron because it is motor. It's a neuron that's interacting with or stimulating a skeletal muscle. So it's called a motor neuron. And then the muscle part is a skeletal muscle fiber. So the neuromuscular junction that we are talking about right now is between a motor neuron and a skeletal muscle fiber. The neuromuscular junction consists of the axon terminal of the neuron. The axon is the part of the nerve fiber that sends the nerve impulse to an organ or a target. All right. In the axon terminal, we have vesicles that contain a neurotransmitter called ACH or acetylcholine, and that neurotransmitter is going to signal the muscle fiber to contract. The portion of the muscle fiber sarcolemma or plasma membrane that interacts with the axon terminal is called the motor end plate. So motor is referring to skeletal muscle. All right. At the neuromuscular junction, acetylcholine is released from the axon terminal. It diffuses across the synaptic cleft or gap between the axon terminal and the motor end plate. It binds to a receptor on the motor end plate. And then that allows for the action potential to be generated, or the electrical impulse to be generated in the sarcolemma, and that will, of the muscle fiber, and that will trigger the muscle to contract. Once this happens, there is an enzyme called acetylcholinesterase, ACHE, that begins to break down the acetylcholine so that it can be recycled and reused for another nerve impulse. Okay, so this is showing you those steps that I just went through with you. You can see down here at the bottom that here's basically is your neuron, and here are your vesicles containing acetylcholine. So what's going to happen is the acetylcholine is going to, by exocytosis, it will fuse with the plasma membrane of the axon terminal here in blue, and then it will release the acetylcholine into the synaptic cleft. The acetylcholine, which is red, will bind to receptors on the motor end plate. That's what you see here, here, and here, and here. And then that's going to trigger sodium ions to come into the muscle, into the motor end plate, and it's going to trigger an action potential in the muscle. And now we'll talk about the contraction cycle. In the contraction cycle, you have, so what has just happened is sodium has entered into the muscle fiber, and that is going to trigger an action potential that travels along the muscle fiber down into the T-tubules. And once that action potential reaches the T-tubules, calcium will be released from the terminal cisternae. That calcium is going to bind to troponin. Remember, troponin is holding the tropomyosin in place, so it will cover the active sites. Calcium is going to bind to troponin. The tropomyosin will move out of the way, and the active sites will be exposed. So now the myosin heads can attach to the active sites on the actin, and then they can pull the actin in. They can pull the actin fibers toward each other and cause a contraction. When the myosin head pivots, the sarcomere shortens. The actin fibers move closer together and the sarcomere shortens. That's a contraction. Then ATP binds to the myosin head, and that causes the detachment of the myosin from the actin. And that's a single contraction cycle. Here are the steps that initiate a muscle contraction. If you look at it from what happens in the motor neuron and along the motor end plate, here's your axon terminal. Here's your ACH in red. The ACH is binding to the receptors on the motor end plate. An action potential is traveling down the sarcolemma of the muscle fiber, and it's traveling down into the T-tubules, where it stimulates calcium release from the sarcoplasmic reticulum, from the terminal cisternae of the sarcoplasmic reticulum. And then that calcium is going to cause troponin to move out of the way, and tropomyosin to uncover the active sites. And then myosin can now form a cross bridge with actin, and then it can pivot and do a power stroke. In other words, that myosin head will pivot, pull the actin in, the actin filaments in closer together, like you see down here, and that's a contraction. And then the end of the contraction is when ATP binds to the myosin heads, and then they detach from the actin, and the muscle fiber relaxes. How would a drug that blocks ACH release affect muscle contraction? Well, it would stop muscle contraction. ACH is necessary to lead to an action potential in the muscle fiber, which leads to the release of calcium. And so the ACH is like the first step in the muscle contraction. What would you expect to happen to a resting skeletal muscle if the sarcolemma suddenly became very